Welcome to Biotanemy.
Yeast artificial chromosome. Yeast artificial chromosomes, or YAC, are specialized DNA molecules used in genetic engineering and molecular biology experiments in eukaryotic cells like yeast, particularly Saccharomyces cerevisiae. These vectors are designed to facilitate the manipulation, cloning, and expression of genes within yeast. The features of YAC are: 1. Autonomous replication sequence, or ARS, acts as an origin of replication, allowing the YAC to replicate independently to the chromosomal DNA of yeast. 2. Centromere, or CEN, acts as a centromere, binding to the spindle apparatus to ensure that the YAC is accurately distributed to daughter cells. 3. Telomere, or TEL, provides stability to the YAC by preventing end-to-end fusion and degradation of the chromosome. 4. Selectable markers. Uses: auxotrophic markers. Auxotrophy is the inability of the organism to synthesize one particular organic compound required for its growth. It allows the selection of yeast cells that have taken up the YAC. These markers are genes that complement auxotrophic deficiencies in yeast. For example, URA3 for uracil biosynthesis, LEU2 for leucine biosynthesis, HIS3 for histidine biosynthesis, or TRP1 for tryptophan. These markers will allow us to select the transformed yeast cells by growing them in the media that lacks respective amino acids. 5. Multiple cloning sites, or MCS, contains multiple restriction enzyme recognition sites for cloning DNA fragments of interest into the YAC vector. Also, some additional features depending on the purpose are included, such as expression cassettes, which include yeast promoters and terminators to drive the expression of genes from the inserted DNA. Stability features includes elements like additional telomere sequences or other stability-enhancing features to reduce the risk of rearrangement or loss of the YAC. Reporter genes, such as beta-galactosidase or green fluorescent protein, can be used to visually or quantitatively track gene expression or YAC presence. Integration sites, for integrating the YAC into the yeast genome rather than maintaining it as an apisome. Different kinds of yeast cloning vectors are: 1. Yeast episomal plasmids, or YEP. The YEP vectors are constructed using 2μm plasmid, which contains genetic information for its replication and segregation, and had a backbone of PBR322 and PUC19. They have a copy number of 20 to 200 plasmids per cell. They can replicate independently in the cell. Example: YEP13. YEP13 will get integrated into the chromosome at the position of the LEU2 gene. Rare cases: the entire plasmid will get integrated into the chromosome and may remain integrated or undergo a later recombination, resulting in the excised out from the chromosome. 2. Yeast integrative plasmid, or YIP. YIP vectors do not possess 2μm plasmid. Hence, it cannot be replicated independently. Instead, it has to be integrated into the chromosomal yeast DNA for survival. They only have one copy per cell. Example: YIP5, created by inserting the URA3 gene into the backbone of PBR322. 3. Yeast centromeric plasmid, or YCP. It contains yeast centromere, or CEN. The vectors contain autonomous replicating sequences, or ARS, either 2μm in origin or chromosomal. They have one copy per cell, and can replicate in the cell without integrating into the chromosome and are stably maintained during cell division. Different applications of yeast vectors are: gene cloning and expression, functional genomics and genetic manipulation, yeast-2 hybrid system, reporter assay, etc.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.